forget that. Okay, so let's finish up talking about. I will finish up what Dr. Campbell began talking about on Monday, and kind of finish up with a little bit of RNA structure prediction. I say ribosomal RNA structure prediction because that's what it normally is. Well, honestly, this could be a lot of different kinds of RNA, in fact, as well. So, hey, guess what I did that? And before we get into too much more, I just do also want to let you know that there's one more program to install, JMOL. This is a program for visualizing sequence or 3D structures of proteins. So we'll be using that a little bit in lab and talking about that a little bit on Friday in class. Most of what we're going to be doing in otherwise is going to be online servers, and either you doing little jobs on there or having essentially simulate stuff, since it takes like a day to get back. I'm not going to have you like wait a day for your lab, so I'll provide you for what you would have submitted, excuse me, and then kind of give you what the output would have been had you waited an entire day. That's fine. Okay. So let's move on and talk about the last method that we didn't get to Monday in class of predicting protein structures, and that's protein threading. And what this is doing is trying to find the best way to mount a residue or a series of residues onto a protein in such a way that we're using the known structure of another protein. Now this, at this moment, sounds probably a little bit familiar, but hold on for just a moment. And then for each fold or each section of structure, essentially, which remember all we're doing is you have a torsion angle. For every amino acid we have a torsion angle and kind of an angle of the side chain. And so for each of those, we're going to calculate or estimate the probability that a given sequence then can have that structure. And so the nice thing about this is a consequence of us actually having a lot of empirically knowing the structures of lots of proteins. And that similar folding probably at the fairly basic level or even at the level of the entire core of the protein has probably already been found, that something very similar is out there. Now, of course, this won't work on everything for that reason, but it can work on a lot of things. And that by comparing parts to a sequence database, or the sequence to a database of known three-dimensional structures, we can find that and then use some sort of scoring function then to assign how likely is it that this sequence could adopt this structure. Now, like I said, because we're making this assumption that we can actually, or we do have some similar folding patterns that we have already discovered, this is reliant on us actually having found those similar structure patterns. This means that we can't do this with all proteins, but it works at least somewhere approximately, it appears, on at least 80% of new protein sequences. So a completely novel protein sequence, this is going to work fairly well and fairly efficiently on the majority of them, but not necessarily all of them. So this probably seems similar to what Dr. Campbell was talking about on Monday in homology modeling, where what we did is instead of what we're talking about here, we took a sequence and tried to align it to a sequence of another protein in a database, and then infer structure by the homology of the structure to that. So in that case, we're aligning sequence to sequence. In this case, in threading, we are not aligning sequence to sequence, we're aligning a sequence to a structure. We're going to talk more about how we do that, but what that means, and the nice part of this is that we don't have to have sequence homology here. We don't have to have sequences that are so similar that you actually can recognize them as being similar things. This is something that actually is really useful because, again, we can have proteins that are radically different in sequence that assume similar conformations and have similar functions. but they don't necessarily have similar amino acid sequences. And so being able to predict the structure of those is very powerful, and what is really kind of one of the big powers of protein threading specifically. Now, a couple things here is that, or a couple concepts or assumptions that are going into this is, number one, there's a limited number of folds that happen in nature. And that appears to be actually true. There's a limited number of types of motifs that actually do happen in nature, and we can kind of fit these to those. And that certain amino acids have, quote unquote, preferences for different structural environments. And this in itself provides enough information to choose what kind of structure each amino acid or groups of amino acids is going to contribute to. So just by knowing kind of the probability of certain amino acids to contribute to certain kinds of structures is enough information to choose which is the most likely set of structures that is over a certain area of a protein. Let me give you one example of this, and this is for alpha helices. Now, if you remember from cell biology, there are certain amino acids that are alpha helices makers and some that are alpha helices breakers. So for instance, alanine works really well in alpha helices. So is arginine. So essentially what you're looking at here is amino acid, and of course, there's two different ways of denoting it. And then here on the complete right-hand side, you have what's called the helical penalty. Essentially, if you were assuming that this is in a helix, we're going to incur some sort of penalty for that being in an alpha helix. So essentially here, the larger the number, the less likely it is that this amino acid occurs in an alpha helix. The lower the number, the more likely it is to occur in an alpha helix. So what we'll look going down here on the right-hand side, alanine and arginine are actually very likely to occur in an alpha helix. Also come down here, leucine, lysine, and methionine also are very friendly to alpha helices. So you'll tend to see alpha helices with lots of these amino acids. However, if we look at proline, it is way high at 3.16. Proline, again, having the two connections between the side chain, very rigid. It doesn't make those turns very well. Another one that doesn't make alpha helices very well is glycine. These are amino acids that are known as helix breakers. If you have a mutation that turns something from something else to like a glycine or a proline, and then it'll just totally break your helix. So what we can do now is we can look at an amino acid sequence and determine, based on these likelihoods, what is the likelihood that this amino acid sequence could make an alpha helix. And so we actually don't need to know specifically or have a very specific homology between the sequences, but what we can do is we can look at an alpha helix here and an alpha helix here and say, okay, what is the probability that this amino acid sequence could make this alpha helix? And so essentially this is going to be, we have some protein here that we have a previous knowledge of, and we have a core segment here, or a core set of structures that we have that is known for that template. And these are stored in a protein database or a protein structure database. We then have that template that can be removed then from this protein, that we know the template then of the structure. And what we can do is we can take another amino acid sequence and fit it to that structure. Essentially slide this in, imagine here just for a minute that all of these are tubes, and that you have a string of amino acid sequences that essentially you can slide through here and find the place in which it's most likely, or we find the best fit with each of these structures. So we take this single amino acid sequence and we say, okay, hey, we have this alpha helix right here. We can slide this amino acid sequence through to say where's the most likely place that we have a stretch of amino acids of the proper length to make this alpha helix. And say, okay. And we do the same thing with this structure, and this structure, and this structure here. We're essentially sliding them through and finding the alignment that is most likely to make that particular secondary structure. And so we have a library of these core templates. that we can then begin aligning and assessing scores to each of these individual templates. So again, the big difference between here and homology modeling is that we're not aligning these sequence by sequence, we're aligning these sequences to a structure and finding the probability given this sequence that we could form this structure. And then again, so yeah, sequences are aligned to that. And then we're getting a sequence match, but then again, just restating what I just said, a propensity for that sequence to form that structure. So we do that again by sliding kind of these sequences around in there and finding which line up very well or have the highest probability of forming that structure. But we can also do this by opening up gaps in the sequence or sometimes gaps in the template depending on what algorithm you're using. So essentially imagine here if we have, go back here, if we're lining this up, imagine this is 5 or 10 amino acids long. But we have a sequence here that it'll do it, but it's only 9 amino acids long. We can open up a gap in that sequence to align well. Similar to we're opening up gaps in the sequence when we're doing nucleotide alignment as well. Are there any questions so far on that? Okay, so go back here. So essentially one iteration through this then is that given a protein sequence and a single template, what we can get back then is what is the best alignment of this sequence to that template. And this also gives us a total score. Then we can score this as what is then the best, or what is the probability that this sequence could form this structure of this template right here. But then we expand that to be, given a protein sequence and a library of these core templates, a lot of them, we can align them iteratively to each of these, and then say what is the best alignment to any of these in here, and then it can return a fairly close, ideally a fairly close template that accurately models the core of what that structure is likely to be. Once we have that done, we get refinement on that template. So again, we have a lot of peripheral areas as well. So we're going to have a lot of nucleotides that are interacting with the environment, a lot of amino acids that are interacting with the environment, interacting with each other. And so what we can do at this point is we can refine the interactions between, we can refine this the fit to this template by then refining based on interactions. So if we have physical overlap of amino acids, that's bad. We can move those around. We can move side chains to get side chain charges, interactions optimized. Also interactions with the environment or the solution. There's actually quite a few different protein threading implementations. One that's used most often is iTaser. Dr. Campbell talked about this. This is actually a really successful algorithm. It's come in first in 4 of the 5 last CASP competitions. Again, if you remember, CASP is, they set up a competition once every 2 years of a protein that someone is going to actually do the X-ray crystallography and find the actual empirical structure. And they give out the amino acid sequence to a lot of different people or a lot of different lab groups, and they say, hey, go figure this out for us, and then we're going to see how well that aligns with what actually happens. And iTaser has come in first for the last 5 competitions, so 8 of the last 10 years, I guess. For quite a while. The one that they didn't get first in, they got second in, which, considering how many entries there are, that's really good. HH-Prej, Raptor, Fire, there's actually quite a few implementations of threading, and this seems to be, from my not being a complete person that does this all the time, my reading of this and looking around, protein threading seems to be the dominant approach to structure prediction of proteins at the moment. It's used a lot. Okay. And then one other thing I'm just going to talk about. This isn't so much a method as it is a program, but I thought it's worth talking just very briefly about, is foldit, which is essentially an online game for folding proteins. And this follows kind of a novel idea. I mean, not completely novel, but it's following the idea that humans, there's still things that computers don't do quite as well, or it's very difficult to get them to do as well as what humans do. And the idea is that there's still a component of this when we're talking about protein structure prediction. So it's actually designed by a group at two groups at University of Washington, so right here in the state. They decided they're going to make this program that allowed in a game environment people to try to fold proteins and work out the best folding patterns. So in concept, people are pretty good at pattern finding, and that they're going to try to harness that pattern finding to try to figure out a way to actually fold these proteins. And there's several tools in there that actually give you ways of capturing also some of these algorithms as well. So you can do protein threading, or some limited concepts in protein threading in foldit, and other things like this. So they're trying to maximize what algorithms do best and what people do best, and combine them into one kind of ideal way of folding proteins. Well, there was a paper published in Nature that was saying this, for some hard things that we have had a hard time figuring out the folding, is that this is outperforming computer-only methods. However, it doesn't really do well at CASP. So when it's actually like put head to head, it doesn't do super hot. I actually pulled up the last CASP. Where'd you go? Oh, here's the last. Oh, you can't really even see there very well. What if I highlight things? Does that make it better? Yeah, that makes it kind of better. I'm just gonna highlight all these rankings for the last CASP that have one going on right now. Number one up here is Zhang. This is Zhang and Zhang server here. This is iTaser. It's actually run by the Zhang lab. Anyways, so that's iTaser. And then here, let's go find foldit. So we're coming down. We're coming down. We're coming down. We're coming down. Coming down. Right here. 129 out of 143. What's that? Oh yeah. Let's see here. Good for them. Where's Mongolian? Oh yeah, Mongolian here. And actually, some of these are other ones within foldit. Like Anthropic Dreams right here is a team using foldit. And there's a few other ones, but in general, yeah. Not, yeah. Oops, let's go back here. So, let's fold it. Okay. Are there any questions on protein structure prediction? Yes.
Do you know how the computational methods do with like transmembrane proteins? Because I've heard that those are hard to do experimentally.
I'm not exactly sure, to be totally honest with you. And honestly, if something is hard to do experimentally, it might be hard to do prediction-wise as well. However, all that being said, okay. So let me preface all of this by saying, I don't know. Okay? And all of this that follows is just my speculation. My hunch, though, is that, okay, so I can see it going two ways. Number one, if it's hard to do experimentally, since we're essentially pulling off empirically found things, then it could be hard because you have less templates to draw off of. And both homology modeling, essentially everything that we're doing is going that direction. Now you could go ab initio, and that that might work a little bit better. In fact, I could see that if you were solely going off of essentially first principle of protein or side chain interactions. I can actually see it being easier because you have essentially more information. Because you know it is a transmembrane protein. So you know that you have some of this that needs to interact with an aqueous environment, and some of this that needs to be externally interacting with a hydrophobic environment. The transmembrane sections need to be hydrophobic to interact well with the membrane. So I could see that being an additional source of information that would actually make it somewhat a little bit easier, but all that being said, I'm not sure. But good question. Any other questions? Okay, let's talk about RNA structure prediction. And one obvious thing is, like, who cares about RNA structures? I mean, yeah. And there's a couple of reasons. Number one is that in the same way that proteins have structure and then will use or have structure that's intrinsically intertwined with their function, and they do lots of functions, RNA does form ribozymes that is kind of in the exact same situation. It forms structures and then carries out functions. And not only that, it's just good to know. There are other reasons why you would want to know RNA structure as well, but that's one of the most interesting ones, is figuring out structures of ribozymes. And one of the most common ones that we do this on actually is a ribozyme, is ribosomal RNA, which particularly, it's not the transfer RNA that is on nucleotides that we're going to be adding on. It's actually part of the ribosome. It actually turns out, like, what, 60% of your ribosome is not protein, it's RNA. And it's forming this ribosome, particularly the large subunit, just as an example, of the, so if you don't remember, you have two different subunits that of ribosomes are in, or ribosomal RNA that's in the ribosome. You have a large subunit, you have the small subunit, and then you have the messenger RNA is kind of sandwiched in between the large and the small subunit as it's getting fed through. And the large subunit actually catalyzes reaction. It catalyzes the formation of the peptide bonds as we are adding amino acids onto a protein. So this is kind of nice and important. Now, one interesting thing, a nice thing about this, is that this is actually way easier to predict than protein structure. A couple reasons for that. Number one, proteins, you have like 21 amino acids, and they all do slightly different things, and they all pair in very weird and in both strong and weak ways, and all sorts of crazy stuff going on. And nicely, in RNA, you only have 4 different bases, and they interact in very predictable ways. And so, base pairing, the nucleotide base pairing, is way easier to predict than the interactions between amino acids. Grand. And what we have here in RNA is we have our traditional Watson-Crick canonical pairings. When we're talking about, remember when we're talking about RNA, we don't have thymine, we have uracil, so we have A's and U's are pairing together, G's and C's are pairing together. Actually, it turns out U acts a little bit weird, so we have one non-canonical pairing that can happen as well. We can have G and U pairing, which is interesting. So that's cool. And they come together, they create just like what we have as motifs in proteins, like for instance we have alpha helices, we have beta pleated sheets, and these other kind of known, repeated, interesting motifs that occur again and again and again and form kind of our underlying repertoire of what we have for protein structure. We have the same thing in RNA. We have like just your standard helix. That's just a stretch of paired bases, and it by default will form this helical structure. Cool. In RNA, we also have a hairpin loop that has 2 different structures to it. It has essentially this part right here, we have base pairs, and those are pairing, and that is the stem, or the stem of the hairpin. And then we have the loop itself, which is just these unpaired, this stretch of unpaired amino acid, or excuse me, nucleotides up here that then loop around. We have a bulge loop in which we have one side that essentially has some additional base pairs. Here on the non-bulge side, this will be consecutive consecutive nucleotides that are bound or pairing with the opposite side, but then we have a bulge here where they will come out and actually have non-pairing going on. We have an interior loop that essentially we have an area where we have base pairing before, base pairing after, and this bulge in between. We have multi-branch loops where essentially instead of going off and having one entrance and one leaving, we have multiple stretches of base pairs coming into it. We also have one that isn't talked about up here, which is called a pseudoknot, which is kind of interesting as well. A pseudoknot looks a little bit like this. Hold on. I am totally blanking. I was like, oh yeah, I can draw that. That's no problem. Doesn't look big. Let's Google it. Ribosomal RNA pseudoknot. Maybe I should. Here we go. I was kind of getting there. Okay, so essentially what we have here now. Yeah, I'm like, wait. Yeah, three-way, okay. So we come in, we have base pairing, we have a loop, then one end of that loop is then base paired again to the other side, and so you get kind of this S-curve going on. This is an RNA pseudoknot. It's not actually a knot. We're not kind of folding in on ourselves, but it is. So hey, we have these RNA structures, these motifs. And then we can express these in this notation, the structure in what's called dot bracket notation. And so how we do this is generally this is underlay. We have the sequence of like U, U, A, G, C, U, A, U, 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 whatever. And then the dot bracket notation will be expressed below this, where we have dot, 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 bracket, 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 dot, 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 bracket, 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 dot, dot. Actually, that is not, okay, but that's wrong. Like, that gives you an example of it, but here's the thing, is that this is usually, this is a stem, this is the other side of the stem, so the brackets are enclosing the stem. And then what's in the middle here is the loop. These are unpaired. So these brackets, or the parentheses, are denoting paired bases in a stem. And they're facing inwards towards the center of that hairpin loop, or those bases, those paired bases. And so here, this section here, the dots are unpaired bases. So this would be the loop. And then, actually, let me actually do this so this is right. A, U, okay, so G, this would have to be a C here, C, this would have to be, actually, I could say a U, U, and then this would have to be an A. Okay, there, now we have an appropriately bound thing. So you have the loop here, and then this is one side of the stem, this is the other side of the stem, and the parentheses are then, just like you have in our code, you have each parenthesis then can, generally, if you do this right, be unequivocally lined up with another parenthesis. So for instance, this parenthesis unequivocally pairs with that one, here to here, here to here, and here to here. So actually what this would be in structure is, let me make this. So we have U, 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 all U, 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 and then we have an A, G, C, U, and then we have an A, U, 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 and then we have A, wait, did I do that wrong? Yeah, I did that wrong. It should be G. G. I can make this one U, though. Let's make that one U. To illustrate my point. So this is G, and we have U, and we have U, and then we have a U, U. So what we have here is pair, like that. So we have an A bound to a U, a G bound to a U, again, our uncanonical pairing that can happen in or in RNA, then a C to a G, and U and A. Here we have our unpaired loop, and then we have the entrance to this pin and the entrance there to the pin. That's dot bracket notation. Give you another example of this. I ran like a little tiny short segment on an RNA server last night of some of my ribosomal RNA, like a little chunk of it from 12S of one of my octopuses. And here's the dot bracket notation for this entire thing. So what you see here then is U, this G again is unpaired. So you see that this G right here is not paired, and it shows that. And then we have, so what would this be called again? What's this little motif right here? Good, internal loop, very good. And then we have a G, U, A, U, A, C. Then we have our loop here, like that, and then we have the complementary side coming up like this, and then we have all this other unpaired stuff that's like, yeah, let's just throw that into a loop. In truth, there's probably more pairings off here that I chopped off, and it doesn't know what to do with it.
What's the color?
The color on this particularly is, I believe this is the confidence in each of these pairings. So the redder it is, the higher confidence there is in that, in its the part of the structure it is. So you see that the loop is all, the pin and the loop is all fairly high confidence, and we get onto this loop and it doesn't even know what to do with it. There's lots of greens and stuff, that's low confidence. This particular one. And it's because, hey, we don't have anything else to do with it, let's throw it into a loop. We're not really confident it's a loop. It also has the assumption that I gave it the complete sequence, which I didn't. I fooled it. Oh, that wily server. I showed it in class. I only gave it one line of the code, or only one line of the sequence. Okay, so I'm going to talk about only one way in which we calculate RNA structures. This is the most commonly used, or at least if not always what's used, is usually in almost every RNA structure prediction algorithm I've seen is one of the options that you would choose. And it's just the minimum free energy prediction method. And the first thing that we do is we construct a dot plot for each pairwise base comparison. This is going to look somewhat like a distance matrix, where we have every single base on one end, every single base on the other end, and we look for pairings between them. And we use this to identify stretches of pairs in these structures. And so that looks a little bit like this. This is hard to read, but essentially this is the exact same thing. This is the dot plot for this thing. Oh hey, people have birthdays today. So this is the same plot for this. Get out of here. Layla, you should not ruin my class. There we go. Okay, so what we have here now is a dot plot in which we see every time you have a thing that pairs, you get a dot in there. And as you get more and more adjacent to each other, or longer stretches, the pairings get bolder and bolder and larger. And when there's a few other ways of calculating that, but essentially what we can do from this is identify stretches of pairs. So again, this is, we look here, this is our A, well, it's easier to look here because it's reciprocated. So we have our A, U here, and then the next A, U here. So that's the entrance into that stem of that loop, the hairpin loop. We have that one that's non-base pairing here. And here's all the pairings that go along here. So what we can do is from this, identify stretches that are stems in our final structure. And we can begin by then using that to make an initial molecule, actually structuring bases together into our stems, having our loops, and then going through and using essentially how stable this arrangement will make the entire molecule, calculate a free energy based on this. And we want to have low free energy. We want all of our molecules to be at a fairly low energy state so they're very stable. And then we can start making stepwise changes to each of this, and then recalculating the free energy, seeing if that improves the free energy. And much like the heuristic searches that we have, that Dr. Caldwell is talking about, we have some ability then to, even if it makes them all worse, still accept it and keep moving on and making these small stepwise changes to the molecule and seeing what improves it until we find some place that any change that we make makes fairly significant, or it degrades the free energy or raises the free energy of this molecule significantly, and we arrive at hopefully fairly accurate structure prediction for the RNA. That went quicker than I thought it was going to go. So, are there any questions? We will have lab this week. I'm pretty sure. You should probably show up anyways. And we will be back in the lab. But with that, I will say, enjoy your day, enjoy all of this, and we will talk more about structure prediction things and have you probably doing a little bit of structure prediction in the lab this week. Enjoy.
Give or take, of proteins need chaperone proteins in order to fold correctly. And how do we calculate direct structures?
That's why we don't do that. That's one of the reasons, not the entire reason, but that's one of the reasons that going from first principles is not super widely used. Because we don't really know you have to know a lot about protein-protein interactions. And what's happening with the chaperones, what kinds of chaperones that we're using, which by that point is tons of information to have that's really hard to get. So that is one of the reasons that we don't.